We often picture our immune system's first responders as these, well, cellular Pac-Mans, just gobbling up trouble.
Hmm.
But what if one of the key players in that initial defense was actually a tiny molecule, a peptide with a surprising double life?
Yeah, one that's both a fierce protector against invaders and, well, sometimes a major contributor to unwanted inflammation.
Exactly. It sounds almost contradictory.
It does, and what's fascinating here is precisely that duality. It really highlights how finely tuned, how incredibly complex our immune system is. It's constantly walking this tightrope, between defense and self-harm.
Okay, let's definitely unpack this incredible balancing act. Today, on the Deep Dive, we're plunging into the world of LL-37.
Right.
It's an antimicrobial peptide, but it has some truly intricate relationships with our own DNA and RNA.
Absolutely.
We've been digging through some fantastic videos from the Derma Immunology Dr. Naglaa Elmongy YouTube channel.
Mm.
They really shone a light on everything from LL-37's role in this process called mitosis
Which we'll get into.
to how it's made, its structure, and crucially, these complex interactions with nucleic acids.
And our goal for you listening right now is to kind of distill all that information. We want to help you understand this dual nature of LL-37, how this molecule, which is, famous for fighting infections, can also be involved in conditions like autoimmune diseases, all through these interactions with DNA and RNA.
Think of this as your express route, your shortcut, to grasping a really fascinating, maybe slightly confusing corner of immunology.
Hopefully less confusing after this.
Hopefully. So let's start with the star itself, LL-37. What exactly is this molecule?
Okay, so LL-37 is the only member of the human cathelicidin family of these antimicrobial peptides.
The only one.
The only one in humans, yes. And it's really well recognized for its broad spectrum antimicrobial activity. I mean, it can tackle viruses, fungi, bacteria, even those really tough bacterial biofilms.
Wow, okay. That sounds like a true defender. What gives it this impressive arsenal?
A lot of it comes down to its design, its molecular structure. It's a relatively short chain, 37 amino acids that folds up into what we call an alpha helical structure.
Like a spiral.
Sort of like a coiled spring, yeah. And crucially, at a neutral pH, like in our body fluids, it carries a net positive electrical charge +6.
Okay.
And this charge, combined with its structure, makes it amphipathic.
Amphipathic. That sounds technical.
It just means it has parts that like water hydrophilic and parts that like fats or lipids hydrophobic.
Oh, okay. Like soap, almost.
Exactly like that. Think of a molecule that can get along with both oil and water. That's essentially what amphipathic means for LL-37.
And that helps it fight microbes.
Absolutely. It allows it to interact with the fatty membranes of bacteria, disrupt them, but also interact with the watery environments within our body and, other molecules. This dual nature is really key to its sort of Swiss Army knife functionality.
Okay, that makes sense. So where does our body actually make this molecular superhero?
Well, it starts off as a bigger inactive protein. It's called HCT18. And this precursor then gets snipped or cleaved into the active LL-37 form by specific enzymes.
By activating it.
Precisely. And the enzymes involved can actually vary depending on where this is happening. So, for example, you've got proteinase 3 in neutrophils. Those are a type of white blood cell.
Uh-huh.
And then things like kallikreins 5 and 7 and keratinocytes, the main cells on our skin.
Interesting.
And what's also interesting is that our bodies can ramp up LL-37 production in response to certain signals, like vitamin D3 actually, and also general inflammation.
So our bodies can dial up the production when needed. That's clever. And where do we typically find LL-37, sort of, hanging out in our bodies?
It's pretty widely distributed, actually. You find it in various epithelial cells. Those are the cell's lining surfaces.
Like skin.
Like skin, yes, but also the gut, the respiratory tract, even the testes. And it's also present in lots of immune cells, neutrophils, monocytes, T cells, NK cells, B cells.
So it's right there at the barriers.
Exactly. Its presence in so many of our barrier tissues really underscores its role as a first line of defense.
Now, one term that kept coming up in the videos was mitosis. What exactly is mitosis, and how does LL-37 fit into that picture?
Right. Mitosis. It's a fascinating process. It's something neutrophils do when they encounter certain pathogens. They essentially, they die in a very specific way to throw out these web-like structures.
Those nets.
Literally. They're called neutrophil extracellular traps, or nets for short.
Okay.
And these nets are made of decondensed DNA, both from the cell's nucleus and its mitochondria, actually, plus proteins like histones, other granular proteins, and crucially for our chat, LL-37.
Wow.
And it's not just neutrophils. Apparently, mast cells, which we usually think of in allergies, can release similar kinds of extracellular traps too.
So these nets are like sticky nets made of our own cell guts, basically, to ensnare pathogens.
That's a great way to put it, yeah. Sticky nets. And LL-37 plays a really vital role within these nets.
How so?
It binds directly to the DNA strands in the web. And this binding does something important. It makes the net more resistant to being broken down by enzymes that bacteria release.
So the bacteria try to cut the net.
Exactly. Some bacteria, like Staphylococcus aureus, produce enzymes called nucleases, specifically to degrade DNA. LL-37 binding to the net DNA helps shield it, reinforces the structure.
Makes it a tougher trap.
A more effective, more persistent trap, yes.
Okay, here's where it gets really interesting, because the videos hinted this could be a double-edged sword. How?
Yes, exactly. While these LL-37-coated nets are great for trapping microbes, the trouble starts if these nets aren't cleared away properly after the infection is dealt with.
They linger too long.
They linger too long, and the persistent presence of these LL-37 DNA complexes has now been linked to a whole host of diseases.
Like what?
Well, the list is growing. Things like cancer, chronic sinus problems, autoimmune diseases like lupus and rheumatoid arthritis, psoriasis, even COVID-19 complications, atherosclerosis, thrombosis.
Wow, that's quite a list. So the very thing meant to protect us can actually fuel these serious conditions.
Precisely. The very mechanism can become a driver of chronic illness if it's not properly regulated and cleared up. It's a profound shift in perspective, really.
It really is. But hang on, there was another wrinkle, right? Something about macrophages.
Yes, good point. There's another layer here. In macrophages, which are another type of immune cell, sort of the cleanup crew, the uptake and processing of these LL-37 DNA complexes can actually be beneficial. It can lead to the killing of bacteria that have managed to get inside our cells.
Oh, okay, so for intracellular bacteria.
Exactly. So in that specific cellular context, these same complexes actually support our defenses.
So it really depends on the situation, the cell type involved.
The context is everything. It tells us that simply trying to boost or block LL-37 across the board probably isn't a good idea. It could have unpredictable, maybe even harmful effects. We need more targeted approaches.
That makes sense. Now, the videos also highlighted the role of RNA in all this, not just DNA. How does RNA fit into the LL-37 story?
Yeah, this is a really important and still developing area. Initially, the focus was mostly on the DNA within the nets as the main inflammatory trigger.
Right.
But then a really significant study came out suggesting that RNA within those nets, maybe even more than the DNA, can be a major driver of inflammation, especially in severe conditions like sepsis.
That's a key distinction. So how does L37 interact with RNA then? Does it bind to RNA too?
It does. It turns out LL-37 can form complexes with RNA, just like with DNA. And these LL-37 RNA complexes, they can activate specific receptors on immune cells, toll-like receptors, specifically TLR8, and in mice, TLR13.
The immune system sensors.
Exactly, like security sensors. And when these TLRs get triggered by the LL-37 RNA complexes, they signal the cell to produce cytokines.
The inflammatory messengers.
Right. And those cytokines can then potentially trigger more net release, creating this kind of self-perpetuating cycle of chronic inflammation.
So it's like the immune system gets stuck in an on loop.
That's a good way to think about it, yeah. And it doesn't stop there. LL-37 also has this ability to act almost like a delivery service for RNA.
What do you mean?
It can ferry both single-stranded and double-stranded RNA into cells.
All right.
And once inside, that RNA can influence which genes get turned on or off. For example, it can stimulate skin cells or blood vessel cells to produce growth factors, which could be good for repair or bad in other contexts, like fueling certain diseases.
Right.
It can also work together synergistically with double-stranded RNA to boost the immune response via another sensor, toll-like receptor 3, in the cells lining our airways.
And it seems like LL37 can also promote inflammation through RNA in other ways, too.
Yeah.
The videos mention something about self-RNA.
Yes, exactly. LL-37 can help our own RNA, self-RNA, which shouldn't normally trigger a strong response bind to scavenger receptors on cell surfaces, and that binding can also set off inflammatory signals.
So it's making our own stuff look dangerous to the immune system.
In a way, yes. But interestingly, there's a twist. A slightly modified, shorter version of LL-37, sometimes called LL-37-1729 or LL-34, seems to do the opposite.
Oh.
Yeah, when RNA is presented to skin cells along with this modified peptide, it can actually dampen down the production of type 1 interferon, a key inflammatory player. So it reduces inflammation in that context.
Wow, so just a small tweak in the molecule can completely flip its function from pro-inflammatory to anti-inflammatory. That's kind of mind-blowing.
It really is. It shows just how sensitive these interactions are.
So even subtle variations matter hugely.
Yeah.
Now let's dive into autoimmune diseases. The video is really focused on systemic lupus erythematosus, or SLE. What's LL-37's role there?
SLE, or lupus, is kind of a classic example of how this whole system can go wrong. The current thinking is that in people with lupus, LL-37 doesn't help clear the nets properly. Instead, it seems to stabilize these complexes of DNA and other immune-stimulating molecules within the nets.
So that problem of nets sticking around is even worse in lupus.
It appears so, yes, and this sets up a really nasty cycle. Okay, so you have the DNA from netosis bound to LL-37. Then, in many people with lupus, you also have antibodies that mistakenly target their own DNA.
Anti-DNA antibodies, right?
Exactly. These components, LL-37 DNA, anti-DNA antibodies, form complexes that then activate toll-like receptor 9, TLR9, in a specific type of immune cell called a plasmacytoid dendritic cell, or PDC.
Okay.
And activating TLR9 in PDCs leads to a massive surge in the production of type 1 interferon.
And type 1 interferon is a major driver of lupus symptoms, isn't it?
A huge driver. So you see the cycle. Nets release LL-37 DNA, which triggers interferon, which likely causes more cell stress and potentially more net release. It's really challenging when our defense systems turn on us like that.
And it gets even more complex, right? I think the videos mentioned autoantibodies against LL-37 itself.
That's right, another layer. Many individuals with lupus actually develop antibodies that recognize and bind to LL-37.
So antibodies against the defender molecule.
Exactly. And these anti-LL-37 antibodies can, paradoxically, promote even more net formation. So you get this amplification loop. LL-37 stabilizes inflammatory DNA in nets, triggering interferon, and then antibodies against LL-37 boost the very process releasing that DNA in the first place. It just fuels the fire.
That's incredibly complex. And these LL-37 nucleic acid interactions aren't just limited to lupus, are they? You mentioned atherosclerosis earlier.
That's correct. That type 1 interferon production by PDCs, driven by these LL-37 DNA complexes from nets, that whole pathway has also been implicated in atherosclerosis, the hardening of the arteries.
How does that work?
Well, one study showed that these LL-37 DNA complexes can actually stimulate those PDCs to pump out interferon alpha right inside the walls of atherosclerotic arteries.
Inside the plaque itself.
Potentially, yes. And that interferon could then lead to more LL-37 production locally, maybe more net formation, contributing to the plaque growing or becoming unstable.
So it seems like these interactions are popping up in more and more inflammatory conditions. What about mitochondrial DNA, mtDNA? That was another key point.
Right, mitochondrial DNA. So mtDNA normally stays inside our mitochondria, the powerhouses of the cell. But when cells get damaged, mtDNA can be released.
And the immune system sees it as a danger signal.
Exactly. It acts as what we call a damage-associated molecular pattern. It basically tells the immune system, hey, there's damage here, and can trigger inflammation.
Okay, so logically, you might think LL-37 could help here, right? Maybe bind to the mtDNA and help clear it away, reduce inflammation, especially in things like cardiovascular disease where there's cell damage.
That would be the logical assumption, wouldn't it? LL-37 binds it, neutralizes it, helps clean it up. But the research suggests it's, again, more complicated. Counter to that idea, studies have actually found high levels of LL-37 bound to mtDNA within atherosclerotic plaques and also circulating in the blood of mice with atherosclerosis.
So it is binding, but maybe not in a good way.
It seems that way. These LL-37 mtDNA complexes were found to actually contribute to making the lesions worse in these mice. And surprisingly, they were quite resistant to being broken down by autophagy.
Autophagy is the cell's recycling or cleanup process.
Exactly. So instead of helping clear the dangerous mtDNA, LL-37 binding might actually be making it stick around longer, protected from cleanup, and potentially causing more trouble.
That is really counterintuitive. But even though they resist cleanup, can these LL-37 mtDNA complexes still trigger inflammation?
Yes, that's the kicker. Even though they're protected from degradation, they can still activate toll-like receptor 9.
Ah, TLR9 again.
TLR9 again,
Yeah.
setting off those inflammatory responses. One study even showed that if you used antibodies to specifically target and block these LL-37 mtDNA complexes in mice, you actually got less atherosclerosis, fewer lesions.
That strongly suggests they're part of the problem.
It really does. And similarly, in a different context, sepsis, these LL-37 family tDNA complexes were found to make local inflammation and lung injury worse, again, seemingly by stopping the mtDNA from being cleared out properly by autophagy.
Yeah.
It just keeps highlighting this delicate, sometimes paradoxical balance.
It's truly fascinating. A molecule designed to fight threats ends up binding our own cellular bits and potentially making diseases worse. Now, something else the videos mentioned was post-translational modification, specifically citrullination. What's that about?
Right, citrullination, this is a chemical modification that can happen to proteins after they've been made. In the case of LL-37, certain amino acid residues, specifically arginines, which are positively charged, can be converted into a different amino acid called citrulline.
Okay.
This conversion is done by enzymes called PADs, peptidylarginine deaminases. And calcium levels seem to be important for regulating how active these PAD enzymes are.
And does this modification change how LL-37 works?
Absolutely. Citrullination gets rid of that positive charge on the arginine, since LL-37's activity relies quite heavily on its overall positive charge.
For binding to negatively charged things like DNA and bacterial membranes.
Exactly. Reducing that positive charge can significantly diminish both its direct bacteria-killing ability and also potentially alter its anti-inflammatory or, in some cases, pro-inflammatory signaling roles. It can also change its 3D structure.
And this happens during inflammation.
It can, yes, which is a really important point to consider if you're thinking about designing drugs based on LL-37. You have to account for the fact that the molecule might get modified in the very environment where you want it to work.
That's a great reminder that these biological molecules aren't static. They could be changed, modified, and that affects their function. Now, the videos also really emphasize the structural flexibility of LL-37.
Yeah.
How does that play into all these different interactions?
That flexibility seems absolutely key. LL-37 isn't rigid. It can apparently adapt its shape, sort of mold itself to bind effectively to a whole range of different partners.
Not just DNA and RNA.
No, not just nucleic acids, also things like phospholipids, which make up cell membranes, the ACE2 receptor.
The receptor SARS-CoV-2 uses to get into cells.
That's the one. And even proteins like alpha-synuclein, which is the protein that clumps up in Parkinson's disease.
Wow, that's a diverse list.
It really is. It's like a molecular chameleon, changing its form to fit different roles.
Can you give us a couple of specific examples of what it does with those other partners?
Sure. So it's been shown that LL-37 can bind to the S1 spike protein of the SARS-CoV-2 virus, potentially blocking it from entering our cells that way.
Okay.
It also interacts directly with the ACE2 receptor itself, which, as you said, is another crucial gateway for the virus, so potentially interfering with infection through multiple mechanisms.
Interesting. And the Parkinson's link.
That's quite different. Research suggests LL-37 can actually inhibit the aggregation, the clumping together of alpha-synuclein. Since that clumping is a hallmark of Parkinson's, it hints at a potential, maybe unexpected, therapeutic avenue there.
That's incredible.
The videos mention LL-37 belongs to a class of proteins called intrinsically disordered proteins.
Meaning they don't have one fixed structure.
Exactly. They're flexible, and this flexibility often allows them to interact with many different partners and perform multiple functions depending on who they're interacting with. It really underscores this idea that LL-37's effects aren't fixed. They're highly dependent on context, its surroundings, and its binding partners.
Okay, so bringing it all back together then, thinking about netosis and disease again, the videos really reiterated that strong connection, especially in conditions like psoriasis.
Right, where LL-37 complexed with DNA or RNA from nets seems to really exacerbate the skin inflammation.
And in lupus, as we discussed, where LL-37 protects that net DNA, leading to autoantibody production, interferon, more inflammation. That whole cycle.
And we also saw how LL-37, through these complexes, can stimulate those plasmacytoid dendritic cells to make type 1 interferon, but it can also activate memory B cells, the cells that remember past infections and make antibodies, and it can activate macrophages and inflammasomes, leading to the release of other pro-inflammatory cytokines like IL-1 beta and IL-18.
So it's hitting multiple inflammatory pathways.
It really seems to be amplifying inflammation through several different mechanisms in these autoimmune contexts.
And it's not just autoimmunity. The videos briefly touched on other things, like COPD.
Yes, chronic obstructive pulmonary disease. Apparently, LL-37 expression is high in the small airway epithelium in COPD patients, and conversely, in a rare genetic condition called Papillon-Lefèvre syndrome, where people don't produce LL-37 properly.
They have problems.
They have severe periodontal disease, severe gum disease, highlighting just how critical this peptide normally is for maintaining immune balance, even in places like our mouth.
It really drives home how central this one peptide is.
It truly does. I think what's so compelling about this deep dive into LL-37 and its interactions with the nucleic acids is that it just reveals this incredibly intricate dance. It's
Hmm.
clearly far more than just a simple antimicrobial peptide.
Definitely not simple.
No, it's a central regulator, with this potential for both safeguarding our health and contributing to disease, all depending on the specific context, the modifications, the binding partners.
Exactly. It's actually quite surprising when you step back to think that a molecule so vital for fighting off infections can also be so deeply implicated in things like autoimmune diseases and chronic inflammation. It really just underscores the incredibly delicate balance our immune system has to maintain.
It really does. And this raises a really important, maybe provocative question for you to think about.
Mhm.
Given this remarkable complexity, this duality, what might the future hold for therapies that try to target or modulate LL-37 and its interactions?
Yeah, that's tricky.
It is. Think about the potential maybe for treating infections in new ways or for managing some of these really challenging autoimmune disorders. But also think about the huge challenge of achieving that crucial balance. How do you tweak such a complex system without causing unintended side effects?
Definitely a lot to mull over. So thanks for taking this deep dive with us into this truly fascinating and, as we've seen, really complex area of immunology.